Entomopathogenic nematodes are an essential part of a thrips IPM strategy. This video will show you how to check the viability of nematodes and how to apply them. For more information on their biology, consult the greenhouseipm.org website. While there are several species of insect parasitic nematode that are commercially available, Steinernema feltii is the species of choice for thrips management. They actively search for their host and once located will invade the host through natural body openings. Once inside the host, they release bacteria carried in their guts, and toxins produced by the bacteria kill the insect. To be able to locate and infect thrips, they have to be alive and healthy. Dead nematodes will not kill anything. To know if the product you are going to use is viable, or to check if passage through your application equipment has not damaged the nematodes, you can do a simple test. Place a small amount of the product in a Ziploc bag with about 5 milliliters of water at room temperature. Mix to create a suspension and leave for a few minutes to allow the nematodes to rehydrate. Alternatively, take a 5 ml sample from the spray tank, nozzle or irrigation system. After waiting for a few minutes, check to see if the nematodes are actively moving or swimming. To do this, you'll need something to create a dark background, and a hand lens or microscope to see them. As nematodes are less than 0.6 millimeters long, live nematodes hold their body in an S shape, or have a slight J curvature at the end of their bodies. Most will move slowly. Dead nematodes will be straight and still. Research shows that thrips are most effectively targeted during the pupal phase, in the growing medium. Consequently, nematode applications typically target the top of the soil. For this, they are applied directly to the soil through the irrigation system, or are applied as a sprench, which means that the heavy spray penetrates the foliage or drips off the plant and into the substrate. Here you see a light spray. The nematodes end up only on the foliage, where they'll have little effect on thrips. In contrast, the heavier sprench application allows the nematodes to reach the substrate, where they are most effective. Here you see the sprench again. As the plant canopy closes when the plants grow bigger, it's more difficult to reach the substrate with a nematode sprench. This graph shows the effect of the growing canopy cover on the amount of nematodes that reach the substrate when applying them as a sprench every 2 weeks for 6 weeks. If there are no plants, nematodes accumulate in the substrate after each application. When nematode application is stopped at 6 weeks, the number of nematodes in the substrate declines quickly. Now, with plants, the canopy grows and closes over time, covering the entire substrate at about 6 to 8 weeks. You can see that as the plants grow, they shelter the substrate, so fewer nematodes reach the growing medium, and the total accumulation of nematodes is less. When nematode application is stopped at 6 weeks, we again see that the number of nematodes in the substrate declines quickly. Prior to this point, many growers will start to use other biocontrol agents for thrips control, like predatory mites, but used together with nematodes in an IPM strategy, they provide very effective control of thrips. Again, for more details on how to prepare and apply nematodes, refer to the greenhouseipm.org website under Biocontrol Agent / Insect Pathogenic Nematodes.